Today I'm going to tell you about microRNAs and the impact they have on clinical data, on experimental data, and why we should consider them every time we look at different experimental settings. So the title of my talk is microRNAs, New Kids on the Genomic Block. So let's start from the beginning. This is a gene expression pathway, and the central dogma up till about 10 years ago. In one cell, we have one genomic DNA which is transcribed into one mRNA, and the mRNA is translated into one protein. So this one gene, one mRNA, one protein was prevalent for a very long time. However, about 10 years ago, it was apparent that many of the genes generate other RNAs, and these RNAs are non-translated, but they get halted at the RNA level. And what do? They actually bind other RNAs, and once they bind them, they inhibit the protein translation. The largest group of these non-coding RNAs are termed microRNAs. So if this squiggly line is an mRNA and we have the 3' UTR, the microRNA binds the 3' UTR, and the critical region for binding is the 5' end of the microRNA. Upon binding of a microRNA to its target mRNA, the mRNA is either degraded or translation is inhibited. But I want to start with a very basic question. Why study microRNAs? And I have three simple, straightforward answers for that. The first one is that microRNAs are ubiquitous. MicroRNAs are everywhere. Every system you look at, you find microRNAs in your cells involved in different cellular processes. MicroRNAs are also mysterious. Even though extensive research is carried out on microRNAs, very little is known about how they function. And microRNAs are important for your health. Different levels of microRNAs were associated with different diseases, in particular cancer. Also, very importantly, there are currently microRNA therapeutics in clinical phase trials. That means that we will probably see microRNA therapeutics in a few years. So I will divide my talk into three different sections. The first one is microRNA genes. Where do they come from? Where are they expressed? And what can we do with them? The second one is microRNA targeting. After we have the microRNA genes, how do they target their mRNA? And the third one is microRNA roles. Well, after we find the keys, all the microRNAs, and we know how to fit them into the locks, we can open the box here, the treasure box, and look at the functions of microRNAs. So if you bear with me to the third section, you will hear about how microRNAs dominantly regulate gene expression in clinical settings. On each section, I will talk about 10 to 15 minutes. So microRNA biogenesis. Where do microRNA genes come from? Here you can schematically see a microRNA gene drawn here as a loop secondary structure because it binds on itself on the RNA level. It could be intergenic or intronic. Here you can see it is between two different exons. It is transcribed by RNA polymerase II, just like regular genes, to form a pre-microRNA, which is processed by an enzyme called DROSHA to form a pre-microRNA, which is transported from the nucleus to the cytoplasm and then processed by another enzyme called dicer. Dicer generates the mature microRNA, and I want you to remember this enzyme. I'll talk about it later. Then the mature microRNA is incorporated into the RISC, RNA-induced silencing complex. The microRNA binds the mRNA and then leads to facilitated mRNA degradation or translation inhibition. So this is the entire process of the microRNA generation, binding, and regulation of an mRNA. It's a very simple system compared to many other complex ones, such as the splicing reaction, which could involve about 200 different proteins. Here, it's a very simple mechanism, a very simple process of about 5, maybe 10 proteins maximum involved in generating the microRNA, taking the microRNA, binding to an mRNA, and then facilitating degradation of the mRNA. So now we're moving on to the first chapter, microRNA genes. And the scientific question we're asking here is how widespread are microRNA genes? This is the logo of miRBase. This is the microRNA database online, which is maintained by the Sanger Institute in the UK. It was quite apparent to us a few years ago, and still today, that there are some discrepancies in the number of microRNAs. So humans have about 1,000 different types of microRNAs recorded, and several monkeys have about 100 microRNAs. So is this a species-specific gap, or is this just a discrepancy in the database? So we asked here a bioinformatic question, and that is, can we find additional microRNAs by homology searches? So what we did, we downloaded all the databases of miRBase with all the microRNAs of the different species, and we also downloaded all the different genomes available. What we did, we scanned all the human microRNAs against all the other genomes. Also, all the mouse microRNAs against all the other genomes. Once we found a particular hit, a match, we retrieved that sequence from that genome. And after we retrieved it, we looked for microRNA features. So the different features are aligned here, but basically we looked at the folding, the location of the mature RNA, and mostly conservation. So by doing this, we identified several hundreds of new microRNAs, or non-registered microRNAs, that we added onto miRBase, and we submitted it onto miRBase. We found very interesting microRNAs, and I'm showing you one example here. So this is a new potential microRNA we identified with our scans. Here you can see a gene, which happens to be an oncogene, HMGA2. The exons are depicted here by boxes, and the intron is depicted by line. So you can see that the potential microRNA we identified, HSA, which means human miR763, which is an homologue to a mouse microRNA 763, is located within the intron. How do we know if this is a real microRNA? So the first thing we did is we looked at conservation in that intron. You do not expect the intron to have high conservation. We did find high conservation in this intron around the region of the microRNA and also slightly upstream of it. That's 100% conservation of 28 different genomes besides human. Then we looked at the expression of these regions. We looked at ESTs, expressed sequence tags, databases, and we found that this region is expressed at 4 different cancer databases or data sets derived from cancer of the eye. And to make this story even more interesting, what we found is that this sequence, this potential microRNA, has a complementary binding site in the 3' UTR of the same gene, which is conserved between human, mouse, and rat. So what does it mean? The microRNA we found can potentially be expressed together with its oncogene. Once it's expressed, it can potentially also bind the 3' UTR and downgrade it. What does it mean? It means that the oncogene could regulate itself or downregulate itself, and that actually makes sense. It's like taking a drunk driver and putting him inside a car and pulling up the handbrake and telling him, now you can drive. You always want to regulate an oncogene. So this is self-regulation of this particular oncogene. We found this bioinformatically. We have not pursued this further because this was a bioinformatics study. In order to make viewing the total world of microRNA easier, we set up a web server called miRViewer, where you can see on the x-axis the different genomes. We have now about 50 genomes here on the x-axis. Here you can see human, and then you can see the different monkeys all along, and it doesn't even fit in the screen. And on the y-axis, you have all the microRNAs all the way down to the bottom. There are thousands of different microRNAs. Everywhere you have a green box, that means that microRNA is present in that genome. So you can see these are highly conserved microRNAs in 50 different genomes, only missing from these different gray boxes here. LED7 is on top, the most conserved microRNAs, and here you can see miR34 just below it. If you scroll down the list, you see very interesting patterns. The pattern you see here is primate-specific microRNAs, only in human and the different monkeys, lacking from all different organisms. These are presence and absence of microRNAs in a particular genome. It doesn't mean anything about the expression. So what we're doing now is we're scanning different cell lines, different systems to see whether microRNA is not only present but also expressed. How do we do it? We do it usually with chips, with microarrays, or we did it up till recently with chips, meaning that you squeeze out the RNA, you label it, you pour it on a slide with antisense against all the microRNAs. It binds overnight. This is how the chip looks like, and in the morning when you come and scan it, you have a whole world of expressed microRNAs. Everywhere you have a dot, you know that that microRNA is present there. However, very recently we moved on to another technology, and that's deep sequencing technology. Deep sequencing allows us a huge leap in our ability to look at the different sequences and to scan the different sequences. This is the machine we use, the Illumina deep sequencing. And metaphorically, I can say that instead of drinking our RNA from a dripping tap, we moved on to drinking our RNA from a fire hydrant. It's that much of a leap. And if we look at the numbers, with the old sequencer, we would read about 100 reactions, 20 nucleotides each microRNA. That's about 2,000 nucleotides. With the new sequencer, we read about 10 million sequences of 20 nucleotide microRNAs. So that's about 20 billion reads every time, nucleotides every time we read, we have a throughput through our machine. That's a 10,000 gap, 10,000-fold increase of reading sequences of microRNAs. How do we use our protocol? How do we read sequences in our deep sequencer? What we do is we squeeze again our small RNAs from our cells. We add linkers. We size select it from a gel. We take only the 20, 30 nucleotides out of the gel, and then we place it on our machine and read those sequences. In order to deal with a very large amount of data of the deep sequences, that 10,000 times fold more increase in nucleotides, we wrote another software called miRNA, freely available online, and you can download it. What we wanted to do is we wanted to allow many researchers to be able to use deep sequencing data to look at their different microRNA levels and also differential levels of microRNAs between two different samples. So this is the input page of miRNA, and you can run it on your local machine, and this is the output page. What you can see here on top, for example, is miR21 highly expressed, so you could read it in one sample 750,811 times. miR21 was read in one sample, and in the other sample it was read slightly less, and now you have your fold change and your p-value. So we have a software allowing many researchers to use deep sequencing data to analyze their different runs. And now on MirViewer, on our global view of microRNAs, you can fly over the microRNA world, look at the different microRNAs of interest you're working on, look at the different organisms, at their conservations, and with an additional layer of expression which we're adding in the next couple of weeks. So I told you that we use deep sequence. We moved on to using deep sequencing. And one very astonishing thing we found is that when you squeeze all the small RNAs out of the cell, you size select them on a gel, you extract the 20, 30 nucleotides, and then you put it on the deep sequencing, you find that about 50%, up to 50% of the small RNAs in your cells are microRNAs. 50% are microRNAs. So first of all, it indicates how important microRNAs are. 50% of all the soup of small RNAs in your cells are microRNAs, meaning they have a very dominant role. Equally surprising is that 50% are not microRNAs, meaning that there are still many small RNAs in there that can probably have a regulatory effect on different genes or different cellular pathways. So to deal with the other RNAs, other than non-microRNAs, we wrote another software, R&A, where here you don't only compare to the microRNA database, but you can compare to many other databases of small RNAs. Currently, we're running many experiments on small RNAs, and I can tell you that when you're comparing diseased samples to normal samples, it could be any disease, cancer, or neurological disorder. When you compare different levels of small RNAs, we find a huge magnitude of difference between the population not only of microRNAs, but also of tRNAs, of SNO RNAs, and ribosomal RNAs. Now, does that have any meaning for diagnostic or therapeutics or mechanistically? We still don't know. But what we know is that now we have tools to profile the entire set of microRNAs and all the other small RNAs. You can see them indicated here. You can profile all of them and try to understand whether you can use these small RNAs as prognostic markers for different diseases. So to summarize this chapter, microRNA genes. We set up a server called MirViewer where you can fly over the microRNA world and look at their conservation. Looking at conservation is extremely important for several reasons. You could look at their conservations if you're looking for a model organism. You could look at their conservation if you're looking at different targets. We'll mention that later on in the talk. You can also look at their conservation to see whether your microRNA has any clinical importance in different organisms or is conserved in different organisms based on their genomic location. Now we're adding different levels of expression on it. Could be based on microarrays, could be based on deep sequencing, and we have added deep sequencing tools, easy to use deep sequencing tools that you can now run on your own samples or even data sets you can download from different databases. So that was the first chapter about microRNA genes. And now we're moving on to microRNA targeting. So after you looked at your microRNA genes, you know its conservations, you know whether it's expressed or not, you want to see where does it bind and what does it bind to. So we're moving now from the microRNA genes from the nucleus. We'll move out into the cytoplasm, and we're going to look at the targeting, at the microRNA itself, and how it finds its targets and binds its targets. So the question in the second chapter here I'm asking is, what determines effective microRNA targeting? Now I'm asking these questions because if you would have come and asked me a while ago, what are the potential targets of LET7 microRNA, I would have showed you a large bag of potential targets, and I would have wished you good luck in finding the right ones because there are many false positives in these samples. So let's look at the interaction between the microRNA and the mRNA, and let's look at the RISC complex. RISC stands for RNA-induced silencing complex. Let's increase its size. Let's peel it from its proteins. Let's change the wiggly lines by real sequencing, and let's zoom in. Now what we're looking here is a zoom in on the interaction between the microRNA and the mRNA potential target. Here you can see the entire sequence of the microRNA, 5' to 3', and here you can see a very short sequence of a very long 3' UTR and mRNA. So this is only partial sequence of the 3' UTR of the mRNA. Here you can see the label is 1 to 8, the 5' end, and here you can see the label is 1 to 9. Very early in microRNA research, it was apparent that the critical region for targeting, functional targeting, is the 5' end of the microRNA. Now what did researchers do? They took these microRNA and then they scanned the different 3' UTRs and looked for a match. Now once you found a match between the microRNA and the potential target, and that target was conserved between human, mouse, rat, and dog, between at least 4 organisms, this region, which was termed the seed region, was rendered the critical site for regulation, and this target, this mRNA, received a flag. This mRNA was a potential target of this microRNA. This was the only way to predict a microRNA target for quite a while. This started in 2003 at MIT from the lab where I came from, and then it advanced onwards with different features. However, the critical question was, what determines effective microRNA targeting? Targeting is not enough. You could find your target, but you might not potentially downregulate it. What determines effective targeting? So we took an experimental procedure here, an approach, and what we did is we took a different cell line and we engineered it to have a dicer knockout upon induction of a particular chemical. So upon addition of a chemical into this cell line, we induced dicer knockout. When you don't have dicer in your genome, you don't have microRNAs. We also used transfection of different microRNAs. We extracted the RNAs from these cells with and without the microRNAs. We used microRNA expression arrays, mRNA expression arrays, and then we analyzed the data. So the critical point here to show you in the experimental procedure is that we can change the levels of microRNAs, either increase them or decrease them, and then look at the mRNAs. We expect a direct link between the changes of microRNAs and the levels of mRNAs. So we profiled our microRNAs again on our chips, on our microRNA arrays, figuring out which microRNAs increased or decreased based on either transfections or dicer knockouts. We also profiled our mRNAs, the entire set of mRNAs, on commercially available arrays, and these are the gene chips of Affymetrix. So here we could look at 20,000 genes, which one did not change, which ones increased, and which one decreased. Now what did we do with the gene expression data? It is an extremely powerful tool. We took the 20,000 genes, and from these gene sets, we took out a few hundred mRNAs that each one has one binding site for the black microRNA, for this particular one. We took another set of a few hundred mRNAs that each one has a particular binding site for the red microRNA. So these could potentially be 2 different microRNAs. it could also be two different, for the same microRNA, two different binding sites. Could be a 6 nucleotide binding site versus a 7 nucleotide binding site. It could be that these ones were not effective and these ones were effective. So this gives us the power to look at a group of mRNAs with a particular binding site and ask what is different between these two groups that allows targeting of one and not the other. Another set we can take, for example, is exactly two binding sites for the black microRNAs. So we can not only ask qualitative questions, but also quantitative questions. What is the difference between regulation of this set versus this set? So the first question we asked is, did our dicer knockout cell work? And here we took 50 random sets. We took 50 random sets to see whether we have an effect once we remove dicer. And here on the x-axis you can see the median log fold change of treated, removing dicer versus non-removing dicer. And here you can see cumulative density of mRNA sets. These are the random sets with and without binding sites. Now what you can see here, these are the strongly detected microRNAs following dicer knockout. You can see a shift to the right-hand side. So what we have here is de-repression. We remove dicer, we remove the microRNAs, and we have de-repression of the sets, the different sets of the microRNAs. And here you can see three of them indicated. This is a set of miR19, a few hundred mRNAs, LED7, and miR106. So first of all, it means that our experiment works. It also means that we could change the level of microRNAs and look at the mRNA set to figure out what are the changes, which direction it went. So then we moved on to asking many quantitative and qualitative questions. I'm only going to show you one or two of them here and then tell you the conclusions of our findings. The question we asked is there a quantitative effect? So we took mRNAs with exactly one binding sites, two binding sites for microRNAs, 3, 4, 5, and 6 and more microRNAs. And here you have the mean log fold change of dicer knockout versus control cells. So we were very surprised to see that on a semi-log graph, we have a linear effect here, meaning that every time, at least between 2 and 5 binding sites, every time you increase the number of microRNA binding sites or decrease the number of microRNA binding sites, you have slightly more repression or de-repression with accumulating microRNA binding sites. This was not only true for our system, but also for transfections in HeLa cells and also for a different organism, for zebrafish. So if we translate it to our findings, what do we say? We say that microRNAs can fine-tune their targets. So microRNAs are not an on-off switch. They don't bind their target and shut it off, but rather they are fine-tuners. You can slightly increase or slightly decrease your microRNA binding sites and you'll get slightly more or slightly less mRNA levels. So with that, we went on searching for particular sequences, for particular target sites that facilitate these targeting or effective targeting, and we have found a bunch of them. None of them were specific sequences. I have to tell you that these were general features. For example, open secondary structure was a very important feature. AU richness was another feature. There wasn't a specific sequence, but there was a cloud of different features that we could all combine together in order to figure out which leads to more effective regulation versus non-effective regulation. So we summarized all of our findings in a web server called TargetRank, where you can now look at the different effectiveness of targeting, not only of a particular microRNA, but a set of targets. Now, since then, a few other sites have established based on very similar features than ours, meaning that you can use TargetScan or TargetRank to figure out whether you have effective or non-effective targeting. So it's not that now that you have a large bank of potential at 7 targets, but they're actually ranked from the most effective on top, based on our experimental data, to the least effective at the bottom. And working with a ranked list, it's much easier than just using a very large bank of non-ranked targets. So to summarize this chapter, microRNA targeting, we found new targeting determinants based on our experimental setting, increasing and decreasing the microRNA levels and looking at the set of mRNAs. We found different features implemented in our algorithms now, allowing ranking of different targets. We also found that microRNAs do not function by binding and shutting off their targets, but more likely they can bind their targets and finely tune it, bring it to a perfected level, slightly increasing or slightly decreasing the mRNA levels. And now, after we looked at the microRNA genes and the microRNA targeting, and we have a potential tool for ranking the targets, now we're going to look at the different roles of microRNAs in 3 different stories I'm going to tell you about. The first story is the microRNA roles as an oncogene regulator. The LED7 microRNA is the first microRNA that was identified, and hence it is the only microRNA that doesn't start with a mir and number, but rather it's called LED7. It's highly expressed in different developmental settings. It is during development, it's highly increased in organisms such as C. elegans and even in mice. LED7 is maintained at high levels during the adult lifespan in many of the different tissues. However, it was also found that LED7 is highly reduced in different cancer types, and that is actually the most common microRNA in many cancers to be downregulated at very early stages of neoplastic transformation. Now, this LED7 was associated with one major gene, and that's the same oncogene I told you before, HMGA2. It was actually shown that when you perturb, when you disrupt the binding of LED7 on HMGA2, you facilitate cancer in a mouse model. But given we have a tool now to rank the different targets of microRNAs, we could look at the most effective and least effective. And also we know how microRNAs work. They don't usually go and bind one target, but they rather have a widespread effect that can affect a large set of targets and possibly even a pathway. The question we asked here is LED7 an oncogene regulator? Does it regulate only HMGA2, or does it regulate a set of oncogenes, a large set of oncogenes? So this is our target ranked targets, LED7 targets. After we rank them, it's not a bag, it's ranked from most effective to least effective. If we look carefully at this list, we see that at the top of the list, we have 3 oncogenes, or those that are expressed highly in cancer versus non-cancer cells: HMGA2, IGF2BP1, and Lin28. So we used a few data sets, and we crossed these data sets to see whether we can find this set of oncogenes regulated by LED7. The first data set we took is target-ranked predicted LED7 targets. We crossed that with another data set, and that's downregulated embryonic genes when we know that LED7 goes up. So LED7 goes up, these genes go down. And the third set is a set of downregulated genes after transfection of LED7 into 2 different cell lines. In the center, we received 12 different genes, and very interestingly, 8 of them are associated with cancer, meaning that they're either increased in cancer versus non-cancer cells or known to be an oncogene. Now, we didn't stop at that. We performed another experiment. In our dicer knockout cells, we knocked out dicer for a few days, actually for 4 days only. And then we extracted the proteins from the dicer knockout cells and the non-knockout cells, the control cells. And then we sent it for proteomics. The question we asked is, which proteins are highly expressed when we perturb, when we change the levels of dicer just for a few days in primary cells? Now I'm going to show you the top of the list, and in the top of the list here, you can see on the right-hand side, the most upregulated proteins after reducing dicer levels for 4 days only are these. And within this list, you can see HMGA2 and IGF2BP1, which came up with our cross-section from this data set. So what does it mean? So as a scientist, we were very happy to see these results, but as a human being, it was very concerning. Why was it concerning? Well, all we did is we took primary cells from a mouse, changed the levels of dicer for 4 days. On a Western blot, we could still see dicer. Us human, we have only one dicer gene. Now you can imagine perturbing the levels of dicer genes It could be by stress. It could be by a mutation in the promoter. It could be a mutation. It could be a methylation of the promoter. If you change the level of dicer only for 4 days, and you look at the upregulated proteins out of 1,000, you come up with 2 oncogenes that can potentially cause a cancer in your cells. Now, obviously, this was a very short-term study. We did not check downstream what happens to the genes. It could be, of course, that these proteins are downregulated somehow, are sent for degradation. But in these cells, in our experiments, after a few days, these genes were highly upregulated. These proteins were highly upregulated, and we were very encouraged to see that there's an overlap between the proteomics and the different cross-sections of the mRNA data. So to conclude this study, we found a master regulator, LET7, as a master regulator of a few oncogenes, at least 8 of them that we identified in a cross-section study. Besides HMGA2, we also confirmed the binding of LET7 on IGF2BP1, and we're in the process of looking at the other ones as well. We also conclude that LET7 prevents the re-expression of a set of oncogenes and not only the HMGA2, the one gene that was known to be regulated by LET7. We were very encouraged by our cross-section studies. So what we do now bioinformatically is cross-section many other data sets. So which data sets we take? We take, for example, all the data sets of single nucleotide polymorphisms, different polymorphisms in our genome. We cross that with different sets of oncogenes. It could be oncogenes, it could be tumor suppressors. And the third cross-section is microRNA targeting. So what we receive here in the center is different oncogenes or tumor suppressors that can evade microRNA targeting by changing one nucleotide in their 3' UTR. We have very interesting genes here in the center, potential ones, which we are now testing in our lab. Obviously, other sets we can take are related to other diseases, so we also have sets of neurodegenerative diseases, sets of genes like that. We looked at polymorphisms, and we're crossing that with microRNA targeting and looking at either gaining or evading microRNA targeting by polymorphisms in the 3' UTR. Now, this study leads me to the second story, and that's the story about IGF1R regulation, and this is carried out together with Professor Eitan Friedman, who is a clinician at Sheba Medical Center affiliated with our university. This is a story about insulin-like growth factor receptor, the receptor of IGF1R. Upon activation, IGF1R facilitates many different pathways, whether it's apoptosis, protection, proliferation, transformation, and so on. Probably the most important feature is that IGF1R is highly overexpressed in most malignant tissues, and it has an anti-apoptotic effect. It's kind of a gate that many cancers have to undergo for rapid proliferation. So what I'm showing you here is a Kaplan-Meier plot. On the x-axis we have times in years. You can see a very clear separation here, and here you can see the fraction. So what is the difference between the two groups? The difference is one nucleotide change in the 3' UTR, and actually the data refers to the age at which BRCA mutation carriers were diagnosed with breast cancer. You can see there's a very clear separation here between the two groups. One group would will be diagnosed with breast cancer up to the age of 46, 100% of the females here, and here 60% of the females will not be diagnosed at the same age. I'm not just showing you this, I'm bringing this to you because we identified that this difference in one nucleotide is in the 3' UTR of a potential binding site of a particular microRNA. So bioinformatically we identified that this change in the 3' UTR could be segregated by one binding site that can abrogate the binding of a particular microRNA. Obviously, this was very exciting, and we wanted to prove it also in the lab, so we ran back to the lab and we performed the following experiments. We generated different cell lines. We took IGF1R with the GA, with the AA, and then we generated a third control one with ACT. What does it mean? Here you can see the microRNA, and I told you that the 5' end of the microRNA is critical for regulation. The GA does not bind the U here. The G does not bind the U. The the A nucleotide would bind the microRNA, meaning that the AA version would be regulated by this microRNA, because it can bind the seed region. This would not bind, and obviously the 3 nucleotide mutations we generated will also abrogate the binding of that particular microRNA. So we took this to a luciferase assay. Basically what it means is that we could very accurately measure the protein levels if we increase or decrease the microRNA levels. So these are the results of the luciferase assays. What you see here, that upon binding of the microRNA to the A version, you have 50% reduction in protein expression of IGF1R. With a control sequence, ACT, and the G nucleotide, which prevents binding of the microRNA, you have double the expression, meaning that one nucleotide change can disrupt the binding site of that particular microRNA. But we didn't stop at that. We know that you need a microRNA and the gene for proper regulation. It wouldn't help if you have this gene without the microRNA there. So we actually went back to the pathology department and we looked for those particular slides of these females and many other ones. We took those slides back to the lab, and those were from about 13 years ago, even before microRNAs were identified. These slides were already stained. So what we could do is we could take that slide and we can divide the normal tissue from the cancer tissue. So we did that with a scalpel, and we divided it into 2 different tubes. And we wanted to see what is the level of this particular microRNA in these cells. And what we found is, we found that in the normal tissues from those slides, the miR-515 is highly expressed. In the cancer tissue, the microRNA levels is very low. So what does this actually mean? This means that this could be a dual mechanism for reducing the level of IGF1R or evading the regulation by a particular microRNA. It means that in order to reduce IGF1R, you can either change one nucleotide in the 3' UTR. If you change one nucleotide in the 3' UTR, you get a reduced level of IGF1R expression. However, that's not the only mechanism. The other mechanism could be to shut off miR-515 expression. If you shut off miR-515 expression, you'll get higher levels of IGF1R of that particular gene. We still don't know if this is a mutually exclusive mechanism or an additive mechanism. We're testing these days to figure out which mechanism is more dominant, or maybe both of them are necessary. But what we know is that we identified that microRNA targeting evading can take 2 routes: target mutations in the 3' UTR or microRNA silencing. We're also testing the method by which the microRNA is silencing, though we probably predict that it's methylation of the promoter of that particular microRNA. Another thing we're testing now is to see how does IGF1R link to BRCA1, and we assume that it's possibly through P53 and SP1 through feedback loops proposed a few years ago by a professor at our department. And now to the final story. And this is using small RNAs for pathogen detection. So since we started using deep sequencing, we not only look at microRNAs, but we also look at all the other small RNAs in our cells. And this is what we found when using the deep sequencing and looking at different experiments of infections. So how can we use small RNAs in deep sequencing for pathogen detection? What we did is we took different cell lines in our lab, and we infected those different cell lines with different viruses and different bacteria. After infecting the cells and waiting a few days, we extracted all the small RNAs from the cells like I showed you before, and then we ran it on the deep sequencing. After running it on the deep sequencing, we took all those sequences, and usually we get about 30 or 40 million short read sequences. We align them onto the human genome. So we want to see which ones stick to the human genome. The ones that bind or stick to the human genome, that's wonderful. And usually we perform a human-centric experiment, meaning that we look at the expression of the small RNAs from the human genome. But in this particular experiment, we put that aside. And we looked at the remainder of the sequences. And that's an additional 5 to 10 million short sequences, which are usually discarded, non-used. However, we refused to discard them. And what we did is we ran them against the NCBI database, which includes all the known pathogens and even sections of it. So there are thousands of different pathogens in the NCBI database, whether it's viruses, bacteria, it could be worms, parasites. However, we were not able to identify a very clear hit. And why not? Well, we're working with very short sequences. And if you look for 50 nucleotide sequences in the entire NCBI database, obviously we will receive many hits. So that wasn't enough for us. What we did is we devised an algorithm for assembly of those sequences. We asked, can we take that bag of very short sequences of 15 nucleotides and 20 nucleotides and assemble it into larger sequences by overlapping the different regions? And once we overlap the different regions and reach sequences of about 100 or 200 nucleotides, we were able to find a very clear match for the particular pathogens we used. So which is the data, what data we received when we compared, for example, HIV infection, non-infection and infection? We found that in the non-infection, only 6% of our reads were unmapped. The rest were mapped to the human genome. In our infected cells, 17% of them in the HIV positive were unmapped reads, indicating that we have a lot of reads not mapped to the human genome and we could use them for pathogen detection. After assembly, in the non-HIV infection, we received only 16 contigs or larger sequences of regions, and in the HIV infected, we received more than 800 sequences, more than 800 assembled locked sequences. The most relevant sequences were the ones above 19 nucleotides assembled, which allowed us a very clear hit, and we took a very stringent E-value in our BLAST searches. We were able to rank the different pathogens, and here I'm showing you the ranked hits from our HIV infection. So surprisingly, actually, the top two hits are mycoplasma in our cells that we identified. The one below it is HIV, the exact strain we performed the infection with. Obviously, later on, we also confirmed that we have mycoplasma in our cells, but what we showed here is a co-infection. And we called our method pathogen detection using short RNA deep sequencing, subtraction and assembly. So you sequence the very short sequences, the 15 to 20 nucleotides. You subtract the human genome, and then you assemble those short sequences to receive very clear hits of your pathogens. We now tested it on several different tissues, and we still identify not even RNA viruses, but also DNA viruses. We can now reassemble almost the entire genome. Now we collaborate with different clinicians to receive clinical samples and to identify in clinical samples, for example, in very young children with high fever, whether we can identify the different pathogens in their blood or in their urine and reassemble and figure out within a few hours what are the different pathogens they are infected with. So what did I talk about here today? I talked about the different microRNA genes, and I showed you three tools: MirViewer, where you can fly over the microRNA world and look at the different microRNA genes. I told about miRNA and RNA, which allows scientists to look at different deep sequencing data on your own computer and look at differential expression of genes. I told you about targeting, different targeting. We have a method of ranking the different targets from most effective to least effective, and there are additional tools out there now ranking the different microRNA targets. And I told you about the roles of microRNAs, whether a microRNA is a master regulator of oncogenes, whether oncogenes could be regulated by microRNAs and then evade that regulation by one nucleotide change. And I also told you about small RNAs that could be used for pathogen detection. And if I show you a visual summary of my talk, microRNAs are important because they are abundant, they are important for our health, and still there's a lot of research to be carried out. microRNA therapeutics is on its way. Deep sequencing is an extremely powerful tool, and we have software out there that can facilitate it. I showed you integrative data we use, integrative data in order to find the particular microRNA or the different sets of targets that are affected by a particular microRNAs. microRNA regulation could be extremely dominant, could segregate between life and death or early and late diagnosis of cancer, and how we use small microRNAs and deep sequencing for pathogen detection. This is me sitting in the center, but I wouldn't be here talking today without the wonderful work of the researchers in my lab, the students in my lab, the Genome Center, which I head at Tel Aviv University, and our collaborators from different hospitals in Israel. And I'll be happy to take questions. Thank you.